In class, we've been talking about cells, and we're going to dive a little deeper today and talk about the cell membrane. The first concept I want to talk about is how the cell membrane is primarily composed of lipids and proteins. The cell membrane is considered to be selectively permeable, which basically means that some things pass through the cell membrane more easily than others would. So if you look over here at this picture, you can see that the red circles are passing through and the pink circles are not. That's an example of a membrane being selectively permeable. You're going to see in a few minutes a picture of a cell membrane that illustrates the phospholipids and the proteins. Those are held together through weak interactions, and those weak interactions cause the cell membrane to have a fluid like model. If you look at this picture, these are the phospholipids. There aren't any proteins in this picture. You'll see those in a few minutes. The phospholipids actually move, so they can move laterally side to side, and they can actually flip-flop back and forth. So this is an example of the fluid mosaic model, and if you think about the bubble lab we did in class and how the bubble was able to move with the straws and you were able to bend the bubble in different ways, that's very similar to how the cell membrane behaves. If you look at these two pictures, you can see some obvious differences. First off, there's lots of spaces over on this side. The yellow pieces here, that is the fatty acid lipid tails, and these happen to be unsaturated, so you can see they have bends in them. Over here, these are saturated lipid tails, so they appear to be more straight. The phospholipid structure we already talked about when we did macromolecules, but the phospholipids are the main component of the cell membrane. So here you can see these are the lipid tails, those fatty acid tails, and the top is where we have the phosphate group. This is another example of it if you wanted to look at its chemical structure. The green here shows those lipid tails, the fatty acids. Here, this orangish red color, that's the glycerol that the lipid tails bind to, and then we have our phosphate group up here. And then this is what the this is what the cell membrane would look like. It kind of looks like a sandwich. There's a layer on top and a layer on bottom. And then if you remember correctly, whenever we talked about macromolecules, we talked about how the head, where the phosphate is located, is hydrophilic or water-loving. And then down here we have the hydrophobic lipid tails, and they do not like water, so they hide on the interior. If you look right here, we have this orange and red. Those are those fatty tails. They hide on the interior of the cell, and hydrophobic molecules can enter the cell membrane much easier than hydrophilic molecules, and we're going to talk about that more in a few minutes. Now this is a little bit different structure of the cell membrane than a picture we have seen so far. If you look at this picture, you can still see those lipid tails in there. The red circles is the phosphate head, but now these dark blue structures, those are proteins. Some serve as transport channels to move materials. That would be this big one right here, the cursor's over. We also have some that can bind with signaling molecules, and then we have peripheral proteins, which are right here. They loosely bind to the membrane surface, and then I have an example as a G protein, and we're going to talk about that more a little bit next week. But just know that these giant blue structures, those are the proteins, and they can appear in different shapes and different forms, and they have different jobs. So far we've covered the phospholipids, and then the proteins. There are 6 major functions of those membrane proteins that I just talked about. We're not going to dive in depth into them right now because we go further into detail with some of these a little bit later. Transport is the first one, and that's the one we're going to talk about today. Another function is for enzyme activity, and then signal transduction. The fourth one would be cell-to-cell recognition, so that's how the cells recognize each other. Intercellular joining, which would be the joining of different kinds of cells, and then attachment to the cytoskeleton. The next part of the cell membrane we're going to talk about is the carbohydrates. So we've covered the phospholipids and the proteins. The carbohydrates are these green structures up here. They're really important in cell-to-cell recognition, so how cells recognize each other, because these structures basically identify the cell as what they are. It can be also important when organisms are developing for their different tissue differentiation. And this is why blood transfusions must be type specific. So if you've ever heard of someone getting a blood transfusion, everyone has their own blood type. And whenever you get a blood transfusion, you have to get a transfusion of that same type of blood. That's because the receptors, these carbohydrates up here, they only will bond with a certain, the blood type that you have. So we talk way more in depth about this later, once again. But it can be really important for blood transfusions. So as I told you, some molecules cross the membrane more easily than others. An example of one of those molecules would be some nonpolar molecules. They cross the membrane easily, and some of those would be hydrocarbons, carbon dioxide, and oxygen. They are hydrophobic, and they can dissolve on the hydrophobic interior of the bilayer. And then that hydrophobic core can halt basically the passage of ions and polar molecules, which are hydrophilic. And so those hydrophilic substances avoid the lipid bilayer by passing through transport proteins. And then if you didn't remember what ions or polar molecules are, those are molecules that possess a charge. We've already talked about osmosis a little bit this year, but one of the most important molecules to move across the membrane is water. And water moves across the membrane through osmosis, and it actually uses special proteins called aquaporins, and they accelerate the speed at which water can cross the membrane. And I have here that 3 billion water molecules per aquaporin per minute cross the membrane, so it's a very large amount. This is the next concept I want to talk about. So passive transport is diffusion of a substance across a membrane with no energy investment. Our first type is called passive diffusion, and this is when a substance moves from an area of high concentration to an area of low concentration, and it diffuses down its concentration gradient. So think of that as going with the flow of traffic. It requires no work to be done, so no energy is used, so we do not use ATP here. And some of the things that can move easily are, once again, those hydrophobic substances, so hydrocarbons, carbon dioxide, and oxygen. And then, once again, it's called passive because we don't use any energy. So those are some examples of molecules that pass easily through the membrane through passive diffusion. The next one, once again, is osmosis. This is when water diffuses across the membrane. There are 3 types of water relationships, and we've already talked about these a little bit in class when we did our grape osmosis lab. The first one is isotonic. That's this picture here. This is when there is no net movement of water, so water is moving into and back out of the cell at a pretty much equal rate. We also have hypertonic, which is when the solution is leaving the cell, and hypotonic when our cell swells because water is entering. In an isotonic solution, I just told you there's no net movement of water. They cross the cell membrane at about the same rate. So you can once again see in this picture that the cells are staying the same size. Water is going in and water is leaving. In a hypertonic solution, the cell loses water to its surroundings. So you can see here, these cells have shrank. That is because water is leaving them. The hyper prefix refers to there being more solutes in the water surrounding the cell. So remember that solutes are particles. So that means here, these cells must have had a high concentration of solutes around them. So the water wanted to leave the cell to move out towards those solutes. And then in a hypertonic solution, the cell will shrivel, but it could also die. Hypotonic is the opposite, so this means that there were fewer solutes in the water around it and more on the inside of the cell. So water enters the cell faster than it will leave, and this can cause the cell to swell, and it could also possibly burst. This is an example in an animal cell. So this is normal. Animal cells prefer to be in an isotonic environment where water is moving into and out of the cell at an equal rate. We've talked about plant cells a lot. Turgor pressure is the pressure of the water inside a plant cell pushing outward against the cell membrane. When a plant cell is turgid, it is in a hypotonic environment, and it's very firm, and this is the state that plants prefer. If a plant cell is flaccid, then it is in an isotonic or a hypertonic environment. We already talked about how hydrophobic substances can easily move through the cell membrane through passive diffusion. Well, here that in facilitated diffusion, this is how those hydrophilic substances diffuse across the cell membrane. They need the help of transport proteins because ions and polar molecules, those charged molecules, cannot pass easily through the membrane. And transport proteins are specific, so they are very particular about the substance that they transport. They can work in two different ways. So the first way is they can provide a channel through which the molecule passes, or they can bind to the molecule and carry them through the membrane. All of the examples we've talked about so far have been examples of passive transport, which means that there was no energy required. So now we're going to switch to active transport, which means that we need to use energy, and that energy is usually in the form of ATP. This is when substances move against the concentration gradient, so think about moving against the flow of traffic, and it moves from an area where there is a lower concentration to a higher concentration. The most common example is the sodium-potassium pump. So if you look over here at the picture, all right, this pump moves only sodium and only potassium. And as you can see, these molecules are charged. So normally they do not pass through the membrane easily. They need help. This is the protein that's providing them help. So it actually moves 3 sodium out of the cell membrane, and then it moves 2 potassium into the cell membrane. And in order to get this protein to move, it needs energy, and that energy comes in the form of ATP. The inside of the cell actually carries its own charge, so it's actually negative, and that's in comparison to the outside of the cell. This difference in electric charge can be expressed in voltage, and it is called the membrane potential. Since the inside of the cell is negatively charged, a positively charged ion like sodium on the outside of the cell is attracted to the negative charges on the inside of the cell. And there are two forces that drive the diffusion of these ions across the membrane. The first one is the chemical force, which is the ion's concentration gradient. And then also we have the voltage gradient across the membrane, which attracts positively charged ions and repels negatively charged ions. And this combination of forces acting on an ion forms an electrochemical gradient. Now I want you guys to take a look at this picture, and you are going to answer the questions provided in the Edpuzzle video. The last concept I want to cover is bulk transport or large transport across the plasma membrane. This occurs by exocytosis and endocytosis. Exocytosis is the movement of large molecules across the membrane, and when this occurs, vesicles from the cell's interior fuse with the cell membrane and expel the contents to the extracellular space outside of the cell. And endocytosis is the opposite. It still moves large molecules across the cell membrane. It's the reverse of exocytosis. This is when vesicles form from the plasma membrane and allows the cell to take in the macromolecules. An example would be the engulfing of foreign particles by white blood cells. So white blood cells fight off foreign things in your body, and so you can actually engulf them by endocytosis.